Hello, dear children, welcome back. I'm Dr. Jisha Joe. In this session, we shall discuss about the different stages of meiosis. In the previous video, we have studied about the different stages of Prophase-I of Meiosis-I. During Prophase-I, we have seen what are the things that are taking place during Prophase-I. During Prophase-I, the pairing or synapses of the homologous chromosomes take place. Then after synapses, exchange of genetic material takes place between the non-sister chromatids of the homologous chromosomes, that is crossing over takes place. After crossing over, the homologous chromosomes move apart. Terminalization of the chiasmata takes place, genetic recombination takes place. By the end of Prophase-I, what has happened? Terminalization of chiasmata has taken place, exchange of genetic material has taken place between the non-sister chromatids of the homologous chromosomes. The nuclear membrane and nucleolus have started to disappear. By the end of prophase, almost disappeared. The centrioles have moved to the opposite poles of the cell and they have started producing the spindle fibers. Now we are moving on to the second stage of meiosis-I, or second phase of meiosis-I after prophase-I, which is the second phase. It is metaphase-I. So we have taken a cell having two pairs of chromosomes, and I am moving on to draw a cell at the metaphase stage having the two pairs of chromosomes there. So that is the cell membrane. That is the cell membrane. By the end of Prophase-I, the nuclear membrane, nucleolus have disappeared. The centrioles have moved to the opposite poles of the cell and they have started producing the spindle fibers. I am drawing the chromosomes there, the chromosomes which we have drawn in the previous class. Okay, that was the first pair of chromosome. The second pair I am drawing. We didn't show any recombination in the second pair of chromosome. Okay, each chromosome will have two arms, which are the chromatids. But in the first chromosome, in last class, if you look at the video, you can see in the first, the one pair of chromosome, we have shown the recombination. That is why I have drawn it like that. Okay, now, so these are the spindle fibers arising from the centrioles. The spindle fibers get attached to the centromere of the chromosome. Okay, so that is the metaphase-I of meiosis-I. That is meiosis-I. That is metaphase-I. So during metaphase-I, the separated homologous chromosomes get arranged or aligned at the equatorial plane of the cell with the help of the spindle fibers. The spindle fibers get attached to the centromere of the chromosomes, and the homologous chromosomes get aligned at the equatorial plane of the cell like that. That is the metaphase stage. You can label the parts, the spindle fibers. These are the spindle fibers. Spindle fibers, that is the centriole, the asters. Centrioles are surrounded by radiating rays which are called the asters. These are the homologous chromosomes. At the equator. Okay. So that is metaphase 1. So remember, in metaphase as in mitosis, here also the chromosomes are aligned at the equatorial plane of the cell. That is metaphase 1. Now we are moving on to the next third phase of meiosis 1, which is anaphase 1. What happens during anaphase? The chromosomes of the homologous pair get separated. Okay, so here they are arranged at the equatorial plane of the cell. Now that is anaphase 1. Anaphase 1, you can see these are the centrioles with the asters there at the poles. Okay, here the chromosomes are separated. They are the two thick arms. Chromosomes are in the most condensed stage during the metaphase. That is why they have been drawn like that. Sorry. Okay, that is the first chromosome on one side, and that is the second chromosome. You can see the color at the tip is different. That is because recombination has taken place there. Okay. Okay. That is the next. That is the second. Just darkening that is all, nothing else is being done. Okay. Okay. So here in the anaphase 1, you can see the members of the homologous pair get separated. In mitosis, what has happened during the anaphase of mitosis, do you remember? During anaphase, the centromere splits and the sister chromatids are separated to the two opposite poles of the cell. Here in anaphase 1 of meiosis 1, you cannot see any splitting of the centromere there. You just observe that the chromosomes of the homologous pair, they get separated. They are moved to the opposite poles by the contraction of the spindle fibers. Okay, so here in anaphase 1 of meiosis 1, there is no splitting up of the centromere as you have seen in mitosis. Here we have seen in metaphase 1, the chromosomes of the homologous pair are arranged at the equator of the cell. During anaphase 1, the chromosomes of the homologous pair get separated and are pulled to the two opposite poles of the cell like that. That is anaphase 1. The next stage after anaphase 1 is telophase 1. You know what happens during telophase. What are the things that are happening during telophase? We have seen that the nuclear membrane and nucleolus reappear, the spindle fibers disappear, the chromosomes which were in the condensed stage decondense to form the chromatin fibers. Cytokinesis occurs. So all these are taking place during telophase here. So that is the telophase 1 stage. Telophase 1. Here you can see the cytokinesis occurring by the formation of furrows. Okay. Nuclear membrane and nucleolus have reappeared. Okay. The spindle fibers have disappeared. Now these are the chromosomes there. Okay, here also so you can see the chromosomes inside the nuclear membrane. And finally at the end of telophase, the furrows have reached the reached the middle of the cell, and the two cells have been formed there. I'm just drawing the final stage of telophase. Furrows have extended to the center, to the middle of the cell, and two cells have been formed. Okay, so that is the first cell and that is the second cell after meiosis 1. So by at the end of telophase 1, two cells have been formed, and as you can see, the chromosome number has been reduced here. Okay, so here you can see only one pair of chromosome. We have started with two pairs of chromosomes in the previous video. When we started with meiosis 1, we took a cell having two pairs of chromosomes. Now you can see that the chromosome number has been reduced to one pair of chromosome. That is why in the beginning we have mentioned that it is meiosis 1 which is actually the reduction division, because the chromosome number is getting reduced to half in meiosis 1. Okay, so I hope that is clear. So that is the end of meiosis 1. and after meiosis 1. So meiosis is not complete with meiosis 1. Meiosis is completed only after meiosis 2. So in certain cases, meiosis 2 will immediately begin after telophase. In some cases, there might be a stage in between meiosis 1 and meiosis 2. This stage between meiosis 1 and meiosis 2 is called as interkinesis. Okay, and during interkinesis, there is no duplication of the DNA occurring. Okay, it is just a stage occurring between meiosis 1 and meiosis 2 in certain types of cells, and it is very short-lived, or it occurs only for a short period of time. Now we are entering into meiosis 2. Okay, so each of these cells will undergo meiosis 2. So that is, so again meiosis 2 has prophase 2, metaphase 2, anaphase 2 and telophase 2. So I am going to draw the cell, the first cell during prophase 2. Okay, these are the stages of meiosis 2 which I am drawing here. Meiosis 2, the first stage is prophase 2. Okay, what actually happens during prophase? That is the first cell. Okay, cell membrane, the nuclear membrane and nucleolus start to disappear, isn't it? That is why they are drawn in dotted lines. That is the second cell. Here also we are drawing the nuclear membrane and nucleolus in dotted lines. Okay, the two cells during prophase 2, you can see the chromosomes there. Okay, here and I had told you that meiosis 2 is similar to mitosis. There is no crossing over or recombination taking place in meiosis 2. It is just as we have studied mitosis. Okay, so here you can see the nuclear membrane, nucleolus disappearing, the centrioles have split and they have started moving to the opposite poles of the cell. Okay, so you can see the centrioles moving to the opposite poles of the cell. Okay, that is prophase 1. Okay, so as I told you, after meiosis 1 there is no duplication of the chromosome taking place. Interkinesis is a short stage. It is a very small period between meiosis 1 and meiosis 2. Okay, and so during prophase 2, just what is happening, the nuclear membrane and nucleolus are disappearing, the centrioles split and they have started to move to the opposite poles of the cell. Okay, so that is prophase 2 of meiosis 2. So it is the second stage of meiosis 2, metaphase 2. So what happens to the first cell? In the first cell we can see, that is the first cell I am drawing, that is the cell membrane. Okay, now by metaphase, by the start of metaphase, this nuclear membrane and nucleolus have completely disappeared. The centrioles have reached the opposite poles of the cell and they have started to produce the spindle fiber. So that is the second cell, okay, at the metaphase stage. They have started to produce the spindle fibers. These spindle fibers get attached to the centromere of the chromosome. So I am drawing the chromosomes there. That is the first chromosome. And the second one, it is the second chromosome. Okay. And spindle fibers attached to the centromere. Spindle fibers get attached to the centromere of the chromosome and they are brought to the equatorial plane of the cell. That is, sorry, it is not 1, it is 2. Metaphase 2. Here also I am drawing the chromosomes. The first chromosome. I forgot to show the recombination there. I'm sorry. Okay. And that is the second chromosome. Okay, and these are also aligned at the equatorial plane of the cell by the action of the spindle fibers. The spindle fibers get attached to the centromere of the chromosomes and they get arranged or aligned at the equator of the cell. That is metaphase 2. Now, as next stage of meiosis 2 is anaphase 2. And as we have seen what happens during Anaphase 2, I told you meiosis 2 is equivalent to mitosis. So here what happens during the anaphase of mitosis? The centromere splits and the sister chromatids are pulled to the opposite poles of the cell. So here also the same thing happens. The centromere splits and the sister chromatids are pulled to the opposite poles of the cell. So that is the difference between anaphase 1 of meiosis 1 and anaphase 2 of meiosis 2. Here in anaphase 1 there is no splitting of the centromere. The members of the homologous pair get separated. There is no splitting of the centromere in anaphase 1. We have not seen any splitting taking place there. It is just the separation of the homologous chromosomes. Whereas in the anaphase 2, as we have already seen, meiosis 2 is similar to mitosis. Here you can see the splitting of the centromere taking place. So these are the two cells. You can see the centriole with the asters. Okay. And the centromere splits. Centromere splits. See, centromere splits and the sister chromatids are pulled to the opposite poles of the cell. See, splitting up of the centromere takes place. Here also I am going to show that. So that is the first chromosome. The second one. See, Okay. Centromere splits and the sister chromatids are pulled to the opposite poles of the cell by the contraction of the spindle fibers. We know the spindle fibers are attached to the centromere of the chromosome. So pulling of the chromosomes take place at the centromere. Okay, so that is Anaphase 2. As I already told you, Meiosis 2 is similar to mitosis. So during Anaphase 2, splitting up of the centromere takes place and the sister chromatids are pulled to the opposite poles of the cell by the contraction of the spindle fibers. Now moving on to the last stage of Meiosis 2, which is it? Telophase 2. Telophase 2. So what happens during telophase? By the end of Anaphase 2, the chromosomes have reached the opposite poles of the cell. The chromosomes decondense or uncoil to form the chromatin fibers. Nuclear membrane nucleolus reappear. Disappearance of the spindle fibers. Cytokinesis takes place. So all these are taking place. So that is the first cell showing cytokinesis. This is the second cell showing cytokinesis. Okay. Okay, this is the first cell. This is the second cell showing cytokinesis. Now the chromosomes have reached the opposite poles of the cell. The nuclear membrane and the nucleolus have reappeared. Spindle fibers have disappeared. Okay, these are the chromosomes there. See, here again. Okay, so that is telophase 2 of meiosis 2. As you can see, the chromosomes have reached the opposite poles. The spindle fibers have disappeared. The nuclear membrane and nucleolus have reappeared. Cytokinesis have taken place. By the end of telophase 1, cytokinesis will proceed and furrow formation will proceed towards the center. Okay, this will extend towards the center, finally forming two separate cells. See, cytokinesis or furrow formation will extend towards the center, resulting in the formation of two cells. So from this cell, two cells have been formed, and from this again two cells have been formed. So at the end of meiosis 2, 4 cells are produced. So from this cell, 2 cells have been formed, and from each of this cell that is produced during meiosis 1, it is these 2 cells which enter into meiosis 2. So these are the 2 cells which I have drawn. Each of these cells undergo the different stages or phases of meiosis 2, and finally 4 daughter cells are produced whose chromosome number is reduced. Okay, so we have started with a cell having 2 pairs of chromosomes, that is 4 chromosomes were there, but now look at the daughter cells produced at the end of meiosis. We have each of the cell carries just 2 chromosomes, or only 1 pair of chromosome is present. So actually the actual reduction division has taken place during meiosis-I. Okay, and meiosis-II is just equivalent to mitosis. And another important thing which you should know is the difference between Anaphase-I of meiosis-I and Anaphase-II of meiosis-II. Remember, in Anaphase-I there is no splitting of centromere taking place. It is just the separation of the homologous chromosomes that is taking place during Anaphase-I. Whereas in Anaphase-II, it is just like mitosis, centromere splits and the sister chromatids are pulled to the opposite poles of the cell by the contraction of the spindle fibers. So at the end of a meiotic division, 4 daughter cells are produced and the chromosome number of the daughter cells is half the number of chromosomes which were present in the parental cell. So that is why meiosis is called as a reduction division, and importance of meiosis is that meiosis ensures the formation of a haploid phase in the life cycle of sexually reproducing organisms. So children, we have seen meiotic division. Here at the end of meiosis, 4 haploid daughter cells have been produced. Okay, so with this we come to the end of this chapter. I hope this session is clear to you. If you have any doubts in what we have studied today, please ask your doubts and give your suggestions in the comment section. Thank you.